Today, I will be talking about ways to prevent Streptococcus mutans acidification to prevent karyogenicity. Although there are signs that probiotics were acknowledged by humans in history, the focus of the study didn't start until around 1886 when Escherich related an infant's digestion physiology to its intestinal bacteria. After him, in 1892, Ludwig suggested treating vaginal infections using microorganisms such as lactobacilli, and in the 20th century, Metchnikoff published a book named The Prolongation of Life, in which he suggested that bacteria help with health and prolonging life in humans. In 1930, Schroeder was the first scientist who successfully isolated and cultured lactobacillus so that they were able to survive in the gastrointestinal tract, and they were named probiotics. Today, probiotics are used to treat and prevent diarrheal diseases and other gastrointestinal tract issues. Dental cavities are the main cause of pain and tooth loss, and they can affect the crown and the roots of the teeth. For almost a century, it was believed that all the bacterial community living on the surface of a tooth could cause caries. To remove all the plaque, brushing the teeth, flossing, and using mouthwash was suggested. However, neither reduced tooth decay, and as the use of sucrose was increased in human diet, further increase in dental decay was recorded. The studies to find the main reason behind tooth decay continued until Streptococcus mutans, also referred to as S mutans, were separated from carious lesions for the first time in 1924. S mutants are anaerobes that break down sucrose as a source of energy, and as a result they produce an acidic environment that demineralizes the surface of the tooth. Usually, the mineral loss is recovered with the saliva. However, if the pH remains low, then it leads to a net loss of minerals and eventually disrupts the tooth structure. This reaction is irreversible, and if not treated, tooth karyogenicity will continue to the root and lead to tooth loss. Researchers have realized that a unique characteristic of S mutants is their remarkable ability to survive acidic environments. They have an acid-induced mechanism, also known as the acid tolerance response, known as ATR. When the pH drops, the rush of protons into the cell membrane will cause four responses. First, the proteins begin discharging the protons to reduce the concentration in the cell. Second, the production of NH3 increases to neutralize the H+. Third, the cell initiates nucleotide excision repair to reduce the damage, and fourth, the cell excretes a global signal to other cells to increase the overall survival rate. Sucrose breakdown is another important characteristic of S mutants that allows them to use sugar as a source of energy and to produce plaque. Since these properties are unique to S mutants, focusing on either one of the features can give us a solution to prevent karyogenicity. In August 2002, Hillman published an article entitled Genetically Modified Streptococcus Mutants for the Prevention of Dental Caries, and he exclusively explained how a group of S mutants were genetically modified to produce an antibiotic that's only fatal to wild-type S mutants. These bacteria were also modified to prevent acid production. The genetically modified S mutants were able to substitute for the wild type, but they did not live long due to essentiality of acidification. Because of the unsuccessful attempt, the proposition to remove the acidification of S mutants was not successful, and because the experiments were costly, the study was terminated. Dental care is very expensive, and the lifestyle of many people limits their ability to take care of their oral health adequately. We all know that oral health and overall health are directly related, and in the dental field, many oral diseases initially begin as simple tooth decay. But because they are left unattended, it later causes further complications. So what I want to do now is to push the studies further and to find better solutions to put an end to dental caries. My proposal will be focusing on altering a characteristic of S mutants and reducing the food source in the oral environment to prevent tooth decay. My first specific aim is to study the acid tolerance response in S mutants and to find the mechanism to block the resistance to low pH. Proton pump inhibitors are drugs that inhibit gastric acid production by inhibiting acid secretion. I was thinking PPI can be introduced to the oral community to prevent
created using Capstone.